7 miles below the surface of the Pacific Ocean, the ground drops so deep that Mount Everest would sink inside it and still be buried under more than a mile of water. The Mariana Trench stretches 1,580 miles long, and the pressure at the bottom is strong enough to crush steel. We thought nothing could live there. We were wrong. We are going to visit creatures that replaced their bones with sand, rock formations that act like living cells, and the deepest human dive ever made, inside a sphere barely big enough to stand in. If you enjoy the journey, subscribe and leave a like. It helps more than you know. Strap in. We begin. Picture the longest drive you have ever taken. Now multiply it by 6. That is roughly how long the Mariana Trench is. 1,580 miles of open wound carved into the floor of the Pacific Ocean, stretching east of a small chain of islands most people have never heard of. It sits in the western Pacific, and if you flew over it at cruising altitude, you would see nothing unusual, just flat, grey-blue ocean in every direction. The Terror is entirely invisible from above. The trench is on average 43 miles wide. That is wider than the entire city of Los Angeles from edge to edge. And the deepest point, a specific location scientists call the Challenger Deep, drops to just over 36,000 feet below sea level. To give that number a shape your brain can actually hold, if you stacked the Empire State Building on top of itself 24 times, the top would still not scratch the surface of how deep this place goes. The ocean floor here is ancient. The rock at the bottom of the trench is approximately 180 million years old. Dinosaurs were just getting started when that seafloor formed. It has been sitting in total darkness, under impossible weight, ever since. The trench exists because of a slow-motion collision happening right now beneath your feet. The Pacific Plate, one of the massive slabs of rock that make up the Earth's outer shell, is pushing against the Philippine Sea Plate. When two of these slabs meet, one has to lose. It bends downward and slides beneath the other plate, dragging a strip of ocean floor with it as it sinks into the Earth. That downward drag carved out the trench over millions of years, and the sinking never stopped. It is still happening today, a fraction of an inch every year, like a slow conveyor belt, pulling the ocean floor into the Earth's interior. That process sounds calm. It is anything but. The grinding between those two plates sends earthquakes rippling outward across the Pacific. Some of those quakes are small. Some have triggered tsunamis that traveled thousands of miles before crashing into coastlines. The trench is an active geological engine, not a passive hole. Scientists gave the deepest point its name from the ship that first measured it. The British research vessel, HMS Challenger, dropped a weighted rope over the side in 1875 and recorded a staggering depth. Decades later, a second British ship returned with acoustic equipment, bouncing sound waves off the seafloor to get a cleaner measurement. Each time the numbers came back deeper than anyone wanted to believe. The trench forms a crescent shape when viewed from above. That curve is not random. It follows the exact line where the two plates meet, bending gently across the ocean floor, like a scar left by something enormous, pressing down from beneath. Most of the ocean floor on Earth is between 12,000 and 18,000 feet deep. The Mariana Trench nearly doubles that. It is an outlier so extreme that scientists treat it almost like a separate world. The rules that govern normal ocean environments start to break down here. Biology bends. Physics gets strange. Chemistry does things it has no business doing. And the deeper you go, the stranger it gets, because the pressure waiting at the bottom of that trench is not just uncomfortable, it is something far more extreme than that. Stand outside and feel the air on your skin. You cannot feel it pressing on you, but it is. The atmosphere weighs down on every square inch of your body with about 14.5 pounds of force. You are built for that. You do not notice it. Now imagine that pressure multiplying. Once, twice, 10 times. Keep going. At the bottom of the Mariana Trench, the pressure reaches over 16,000 pounds per square inch. That is roughly 1,000 times the pressure you feel standing in open air right now. To put a picture to that number, imagine 50 fully loaded jumbo jets stacked directly on top of a single person. Every square inch of that person's body holds that weight. That is what the bottom of the trench feels like. A standard scuba tank, the kind divers use at 60 or 70 feet below the surface, would crumple like a paper cup at those depths. A styrofoam coffee cup sent down on a research line comes back the size of a thimble, every pocket of air inside it violently compressed. Researchers actually do this as a demonstration. They drop cups attached to equipment lines, and when the gear comes back up, the cups are miniature, dense, misshapen little lumps. The ocean squeezed them into a fraction of their original size. Steel itself changes under that kind of force. During the very first crewed mission to the bottom, the steel hull of the descent vehicle actually compressed and shrank by a measurable amount as the pressure built around it. The crew inside heard the metal groan and pop as the walls pressed inward. They kept going anyway. Modern deep-sea exploration vehicles use titanium spheres to protect their crews. Titanium is one of the few materials that can withstand that kind of force without catastrophic failure. But even titanium has limits, and engineers calculate those limits very carefully before anyone climbs inside. The pressure does something else, something less obvious but just as dramatic. It changes the behavior of water itself. At those depths, water molecules are packed closer together than they are at the surface. The water becomes slightly denser, slightly more rigid. Sound actually travels faster through it. Scientists use this property to their advantage, bouncing acoustic pulses off the seafloor. To map it, the speed of the returning echo tells them exactly how far away the bottom is, down to a matter of feet. There is a layer of the ocean somewhere between 3,000 and 6,000 feet down, where the pressure and temperature combine to change how sound moves. Scientists call it the deep sound channel. Sound waves get trapped inside it and can travel thousands of miles without fading. Whales use it to communicate across entire ocean basins. Researchers used it during the Cold War to track submarines. The trench pushes well past that layer, past the point where submarines operate, past the point where any animal with lungs survives, past the point where most materials hold their shape. The pressure is the filter. Everything at the bottom of the Mariana Trench earned its place there by solving a problem that kills everything else. And the solutions these creatures evolved are some of the strangest things biology has ever produced. But before we get to the living things, there is something even more surprising about what the pressure does to the chemistry of the water itself, and why it makes the bottom of this trench chemically unlike anywhere else on Earth. The trench is not just sitting there, it is being made, right now, by a collision that has been running for tens of millions of years, and shows no signs of stopping. To understand what is happening beneath the ocean floor, picture two enormous stone slabs floating on a slow river of hot, semi-liquid rock deep inside the Earth. They move maybe 2 inches per year. That sounds harmless. Over millions of years, it reshapes continents. The Pacific Plate is one of the largest slabs on Earth. It stretches across most of the Pacific Ocean, and it is moving westward. The Philippine Sea Plate sits in its path. When they meet at the edge of the Mariana Islands, the Pacific Plate does not stop. It bends downward and dives beneath the Philippine Sea Plate, angling into the Earth's interior at a steep slope. Geologists call this process subduction, and the zone where it happens is called a subduction zone. As the Pacific Plate descends, it drags the ocean floor with it, pulling it downward like a heavy tablecloth sliding off the edge of a table. That downward pull is what created the trench, and it is what keeps it deep. The sea floor at the bottom of the Mariana Trench is actively moving toward the Earth's mantle. It is not a static feature. It is a geological process frozen in slow motion. The descending plate carries water with it. Seawater gets trapped in the rock as the plate sinks, and as the pressure and heat increase deeper in the Earth, that water gets squeezed out. It rises back up through cracks in the crust, carrying dissolved minerals with it. When it hits the cold ocean water near the seafloor, those minerals crystallize and build structures. This is how hydrothermal vents form, and the Mariana Trench has them in abundance along its edges. The grinding between the two plates also generates enormous amounts of heat through friction. That heat, combined with the chemistry of the descending rock, powers a chain reaction. The plate above gets warped and cracked. Magma pushes up through the weakest points. The volcanic islands that form the Mariana Island chain exist because of this exact process. They are the surface expression of what is happening miles beneath the ocean floor. The earthquakes generated by this collision are not small. The subduction zone beneath the Mariana Trench is one of the most seismically active regions on Earth. When the plates lock together and then suddenly slip, they release stored energy in seconds. That energy radiates outward in waves. In 1896, a massive earthquake off the Japanese coast generated a tsunami that killed over 22,000 people. The same geological mechanism drives that risk across the entire Pacific basin today. The crescent shape of the trench follows the exact curve of the plate boundary. It bends because the sphere of the Earth forces the collision zone to arc. A straight line cannot wrap around a round surface, so the trench curves naturally with the geometry of the planet. The seafloor at the very bottom of the trench is approximately 180 million years old. Most ocean floor gets recycled back into the mantle through subduction before it reaches that age. The rock at the bottom of the Mariana Trench survived because it sat at the leading edge of the plate, always being pushed forward, always staying just ahead of the point of no return. That old rock preserves chemical records of the ancient ocean that younger seafloor has already destroyed. And those records are telling scientists something strange about what the interior of the Earth is actually made of. For most of modern science, the deep ocean was a dead zone. Not theoretically dead, dead by consensus, by agreement, by logic so clean that questioning it seemed foolish. Here is the logic. Plants need sunlight to grow. Animals eat plants. Everything connects back to sunlight at the base of the food chain. Go deep enough in the ocean and sunlight disappears completely. Below 3,000 feet, the water is pitch black. No photosynthesis, no plants, no food chain, no life. It made perfect sense, and it was completely wrong. The first real crack in that assumption came in 1977, far from the Mariana Trench, near the Galapagos Islands. A team of scientists aboard a research submersible dropped to nearly 8,000 feet and expected to find cold, dark, empty seafloor. Instead, their cameras lit up something extraordinary. Clustered around cracks in the ocean floor were dense colonies of life, tube worms the size of baseball bats, giant white clams, pale crabs picking through thick mats of bacteria, all of it thriving in total darkness, completely disconnected from sunlight. The team was stunned silent. One scientist later described it as looking at an alien landscape that had no right to exist. They had found an ecosystem running on a completely different energy source, and the implications were staggering. Sunlight was not required. Life had found another way. That discovery launched decades of research into what scientists now call chemosynthesis. Instead of using sunlight to convert carbon dioxide into food, certain bacteria use chemical energy. They pull hydrogen sulfide and other minerals out of vent water and use the chemical reactions those substances produce to build organic molecules. Those bacteria become the base of a food chain. Other creatures eat the bacteria. Larger animals eat those creatures. A whole ecosystem builds itself around chemistry rather than light. When researchers eventually made it deep enough into the Mariana Trench to study its ecosystems properly, they found the same pattern playing out at far greater extremes. The pressure was 1,000 times surface pressure. The temperature hovered near freezing, and creatures were living there in numbers that nobody had predicted. The discovery did more than rewrite marine biology. It rewrote the question scientists were asking. For decades, the search for life in space focused on finding planets with sunlight hitting liquid water, because those are the conditions life needs on Earth. After 1977, that assumption started to crack. If life could thrive miles underwater in total darkness, running on chemical energy from rock, then the conditions required for life were far less specific than anyone had imagined. Suddenly, icy moons with liquid oceans deep beneath their frozen surfaces looked a lot more interesting. Europa, one of Jupiter's moons, has an ocean sealed under miles of ice. Enceladus, orbiting Saturn, vents water vapor into space from cracks at its south pole. Both are now considered serious candidates in the search for extraterrestrial life. And the reason scientists take them seriously traces directly back to what a team of researchers found in the dark, deep ocean nearly 50 years ago. The trench did not just hide life, it hid proof that life is far harder to stop than we ever thought. And what those creatures are actually eating down there leads somewhere even stranger. At the surface of the ocean, sunlight hits the water and microscopic plants called phytoplankton soak it up and grow. Tiny animals eat the phytoplankton. Bigger animals eat those. Eventually something dies. Its body sinks. That is where the deep ocean gets its food. A constant, slow snowfall of dead material drifting down from the world above. Scientists call it marine snow. It sounds gentle. It looks almost beautiful on camera, these tiny white flakes drifting through the beam of a submersible's lights in total darkness. But it is, in reality, a rain of corpses. Dead phytoplankton, decaying fish, animal waste, fragments of shells and mucus, and whatever else the ocean discards. It falls slowly, sometimes taking weeks to reach the bottom. By the time it arrives at the floor of the Mariana Trench, it has traveled more than 7 miles straight down. Everything at the bottom depends on it. The giant single-celled organisms, the amphipods, the rare fish species that survive at extreme depths, they all depend on material that started life under sunlight, thousands of feet above. The trench is the final destination of the ocean's leftovers. But marine snow alone cannot explain all the life scientists found there. The hydrothermal vent ecosystems run on something entirely different. Hot water blasting out of cracks in the seafloor carries dissolved minerals like hydrogen sulfide. Specialized bacteria consume these minerals directly, using the chemical energy released during the reaction. These bacteria form thick mats on the seafloor around vents. They become food for everything else nearby. This is chemosynthesis, and it is completely independent of the sun. A hydrothermal vent ecosystem could theoretically keep running even if the sun went dark tomorrow. The energy comes from the Earth itself, from the heat of the interior and the chemistry of the rock. Near the Mariana Trench, scientists also discovered a different type of vent system, called cold seeps. Where hydrothermal vents push out superhot water, cold seeps ooze methane and hydrogen sulfide at near-freezing temperatures. Bacteria around cold seeps use those chemicals the same way bacteria around hot vents do. Different temperature, same trick. Life adapted to use whatever chemistry was available. The mud volcanoes along the edges of the trench add another layer to this story. These are not traditional volcanoes that erupt molten rock. Instead, they push out a thick mixture of cold water, mud, and organic compounds from deep beneath the crust. When that material reaches the sea floor, it creates pockets of chemical richness that support their own small ecosystems. Scientists have found bacteria living inside the mud itself, pulling energy from compounds that formed miles underground. The trench is not one ecosystem. It is several, stacked and overlapping, each running on a different energy source, each full of organisms that evolved specific tricks to survive their specific corner of the deep. The marine snow community depends on the surface world. The vent communities depend on the Earth's interior. The cold seep communities depend on ancient organic material locked in rock. And remarkably, these systems do not stay separated. Creatures move between them. Amphipods that scavenge marine snow also visit vent edges. Bacteria drift through the water column. The trench mixes these ecosystems in ways scientists are still trying to untangle. What none of them share is the need for bones, because at this depth, bones are something the ocean simply will not allow. Your skeleton is made mostly of calcium phosphate specifically, packed into a crystalline structure that makes it hard enough to hold your body together. Bone is one of the most reliable biological materials evolution ever produced. It shows up in fish, reptiles, mammals, and pretty much every complex creature on land or in the upper ocean. At the bottom of the Mariana Trench, bone does not exist. Here is what happens. Seawater at extreme depths becomes chemically aggressive toward calcium carbonate, which is the material that makes up shells and certain types of bone. The crushing pressure and near-freezing temperatures shift the water's chemistry past a boundary scientists call the carbonate compensation depth. Below that line, any calcium carbonate that sinks into the water simply dissolves. Shells, coral fragments, the tiny calcium-rich skeletons of surface plankton, all of it gone before it reaches the bottom. The seafloor of the Mariana Trench is coated in a soft, yellowish-brown ooze. That ooze is the remains of millions of years of dead marine life that sank from the surface and then partially dissolved on the way down or immediately upon hitting the bottom. The calcium pieces dissolved, the organic material that resisted breakdown piled up. Layer by layer, over millions of years, it built up into a thick blanket of biological sediment, covering the entire trench floor. Animals living down there had to solve the calcium problem. They did it in several different ways. Some creatures use silica instead of calcium to build their structures. Silica is chemically stable at those depths and does not dissolve the way calcium does. Certain deep-sea sponges construct elaborate glass-like skeletons from silica. The resulting structures are surprisingly beautiful and hold their shape under the pressure without issue. Other creatures abandoned hard structures entirely. Snailfish, some of the deepest living vertebrates on Earth, have skulls and bones that are soft, almost jelly-like. Instead of rigid calcium structures, their skeletons are made of a loose, cartilaginous material that flexes under pressure. Their organs are protected by the flexibility of their tissues, rather than the hardness of a cage. Still, other organisms use a completely different strategy. The giant single-celled organisms living at extreme depths, the xenophyophores, build shells out of particles they collect from the surrounding sediment. They glue sand grains and mineral fragments together using proteins they produce themselves, constructing an external structure from the environment rather than growing one from the inside out. Even the muscle tissue of deep-sea creatures differs from what you find in surface animals. The pressure at the bottom would interfere with normal enzyme function. Enzymes are the molecules that run chemical reactions inside cells, and they have specific shapes that let them grab onto other molecules. Extreme pressure distorts those shapes. Deep-sea creatures produce enzymes with slightly different structures that stay functional under crushing force, the result of millions of years of evolution selecting for pressure tolerance. The trench is, in a real sense, a zone where the ocean strips away the most common materials in biology and forces life to rebuild from scratch, using whatever survives. Silica instead of calcium, flexible tissue instead of rigid bone, environmental materials instead of grown shells. And if the pressure dissolves bones, it does something even stranger to fat, something that, without the right fix, would kill every cell in the deep ocean stone dead. It moves through water at 7 miles down, pale white, almost transparent, with a body that looks more like a melting candle than a fish. It has no real scales. Its skin is thin, slightly loose, and you can see the shadowy outline of its organs through it. Its skull is soft. Its bones are more like suggestions than actual structures. And it lives at a depth that would instantly kill every other fish species on Earth. The snailfish, specifically a species discovered in the Mariana Trench, called the Mariana snailfish. In 2017, cameras on a deep-sea lander captured footage of this fish swimming actively in the Challenger Deep, deeper than any fish had ever been recorded. Scientists watching the footage back in the lab were startled. The fish moved confidently, feeding on tiny crustaceans, behaving like a perfectly healthy animal in its natural environment. Not struggling. The pressure that exists at that depth is the same pressure that would collapse a steel submarine. The fish does not feel crushed because it does not have air pockets for the pressure to act on. Its body is filled with fluid, not air, and fluid does not compress the way gas does. Every internal cavity is liquid-filled. Its swim bladder, the organ most fish use to control buoyancy, is either absent or completely modified. Air-filled swim bladders would implode instantly at those depths. The snailfish simply does not need one. Its skeleton is soft enough to flex rather than crack. Rigid bones would fracture under the pressure. The snailfish evolved a loosely organized skeletal system, where the bones barely qualify as bones, more like flexible scaffolding than the hard framework you find in surface fish. But the most important adaptation is chemical. The pressure at those depths disrupts the function of proteins inside cells. Proteins fold into specific three-dimensional shapes to do their jobs, and extreme pressure pushes those shapes out of alignment. The snailfish counteracts this with a molecule called trimethylamine oxide, or TMAO. TMAO stabilizes protein structure under pressure, acting like a molecular brace that keeps the proteins from collapsing. Scientists have found that deep-sea fish have dramatically higher concentrations of TMAO in their tissues than shallow water fish. The deeper the fish lives, the more TMAO it carries. At a certain depth, the concentration gets so high that researchers believe it becomes chemically limiting, meaning the body cannot pack in any more. That depth appears to be roughly 27,000 feet. The Mariana snailfish lives well below that, right near the edge of what TMAO chemistry can sustain. Below that depth, scientists believe no fish can survive. The chemistry simply breaks down and no known adaptation can fix it. The snailfish lives at the very edge of the possible range for vertebrate life, and beyond that edge, the trench is not empty. The creatures that thrive below 27,000 feet abandoned the vertebrate body plan entirely. They evolved something far more alien and far more resistant to pressure than anything with a spine could manage. One of those creatures is a single living cell, a single cell, and it grows to the size of a softball. A human body contains roughly 37 trillion cells. Each one is invisible without a microscope. The average human cell is about 100 times smaller than the period at the end of this sentence. That is what a cell is supposed to be: microscopic, tiny, too small to see with the naked eye. The xenophyrophore disagrees. These organisms live on the floor of the Mariana Trench and several other deep ocean environments, and they are single cells, one cell, no division into separate parts, no tissues, no organs, a single unbroken cellular boundary containing all the machinery of life, and some of them grow to 4 inches across. That is roughly the size of a softball. A few researchers have found specimens approaching the size of a dinner plate. They are the largest individual cells known to science. The name comes from Greek roots meaning bearer of foreign material, which describes exactly what they do. A xenophyrophore builds its outer structure by collecting particles from the surrounding sediment and gluing them together. Sand grains, mineral fragments, tiny shells of other organisms. It cements these pieces into a hard external casing. Using protein, it secretes itself. The result looks, under a camera light, like a lumpy, irregular blob of sediment sitting on the sea floor, except that the entire thing is a single living cell. Inside that casing is a massive branching network of cytoplasm, the living fluid inside cells. The nucleus, the structure that holds the cell's genetic material, is present in enormous numbers inside a xenophyrophore. Instead of one nucleus, a mature xenophyrophore may contain thousands, all sharing the same cellular space. This is called a multinucleate structure, and it allows the cell to grow to sizes that a single nucleus cell could never manage. Why they grow so large is an open question. One possibility is that large body size in the deep ocean is simply advantageous for collecting marine snow. A larger surface area means more falling organic material gets intercepted. Another theory is that the cold temperature and crushing pressure at those depths slows down cellular division, allowing individual cells to keep growing long past the point where surface organisms would split into two. Xenophyrophores also play an important ecological role. Their bodies and the sediment structures they build create microhabitats for smaller organisms. Tiny crustaceans and worms shelter inside and around them, using the xenophyrophore structure as cover. When a xenophyrophore dies, its casing becomes part of the sediment layer on the trench floor, adding to the accumulating record of organic material on the bottom. What makes them particularly interesting to scientists studying extreme biology is what they reveal about cell size limits. On the surface, the reason cells stay small is partly related to the distance that nutrients and signals have to travel inside them. Large cells struggle to distribute materials efficiently. The xenophyrophore solves this with its branching internal network, threading cytoplasm through the entire structure, like a living root system. This organism exists at a depth where most biology collapses, and yet it not only survives, it grows to sizes that seem impossible by normal biological rules. Everything about it breaks the expected pattern. And if a single cell can pull off something that remarkable, wait until you hear what the trench's smallest and most numerous inhabitants did to stay alive. Because the trick they use is something every one of your cells is doing right now, in a completely different way. Your cell membranes are made of fat, specifically a double layer of fat molecules arranged so that their water-repelling tails point inward and their water-attracting heads face outward. This structure, called a lipid bilayer, is what separates the inside of your cell from the outside world. It controls what gets in and what stays out. It is flexible enough to let the cell move and change shape. At normal body temperature, it has the consistency of a light cooking oil, fluid enough to work, firm enough to hold together. Drop the temperature dramatically and that fat membrane freezes. Frozen fat does not flex. A rigid cell membrane cannot let molecules pass through it properly. Signals get blocked. Nutrients stop moving in. Waste stops moving out. The cell dies. This is the problem facing every organism in the Mariana Trench, where water temperatures hover between 34 and 39 degrees Fahrenheit, just barely above freezing. At that temperature, a normal cell membrane would seize up. The fat would solidify, the cell would stop functioning, and the creature would die. Deep-sea animals solved this problem before scientists even knew it was a problem. The solution is a specific type of fat molecule called an unsaturated fatty acid. The difference between saturated and unsaturated fat comes down to the shape of the molecule. Saturated fat molecules stack neatly against each other, like bricks. When the temperature drops, they lock together tightly and go rigid. Unsaturated fat molecules have a kink in their structure, a small bend in the carbon chain. That kink prevents them from packing tightly. They stay loose and fluid even at very low temperatures. Deep-sea creatures pack their cell membranes with unsaturated fatty acids. The higher the concentration, the more resistant the membrane is to cold-induced rigidity. Scientists studying amphipods collected from the Mariana Trench found that their membranes contain dramatically higher concentrations of unsaturated fats than the membranes of related shallow water species. The trench animals had, over millions of years, tuned the chemistry of their own cells to stay fluid in conditions that would freeze normal fat solid. This adaptation is called homeoviscous adaptation. The organism adjusts the composition of its cell membranes to maintain a consistent fluidity across a wide range of temperatures. Some bacteria can do this in real time, changing their membrane composition within hours of a temperature shift. Larger creatures like amphipods and fish lock in a membrane composition suited to their particular depth during development. The same basic principle is used in the food industry. Manufacturers add partially unsaturated fats to products to prevent them from going solid in cold storage. Deep-sea animals evolved the same chemical strategy billions of years before any food scientist ever thought of it. The combined pressure resistance from TMAO and the cold resistance from unsaturated fats means that a deep-sea amphipod is running a completely different internal chemistry from any animal you have ever seen in a zoo or a fish. Tank. Its cells are chemically engineered for an environment that would kill you in seconds. And all of that biological engineering existed in the dark, completely unknown to science, until two human beings decided to go down and see it for themselves. The story of that dive is one of the most quietly terrifying things any person has ever done. January 23rd, 1960. A research vessel sits on the surface of the Pacific Ocean, above the deepest point on Earth. Hanging beneath it, attached to a float filled with aviation gasoline, is a steel sphere barely 5 feet across. Inside that sphere are two men. There is almost no room to move. No rescue vehicle exists that can reach them if something goes wrong. The plan is to sink 7 miles straight down, touch the bottom, and come back up. No one has ever done it. No one knows for certain that it is survivable. Jacques Piccard is a Swiss oceanographer. Don Walsh is a United States Navy lieutenant. Together, they climb inside the Bathyscaphe Trieste and seal the hatch. The vessel begins to sink. The descent takes nearly 5 hours. Piccard and Walsh sit in near silence, watching instruments and listening to the sounds of the vehicle around them. As the depth increases, the pressure builds, and the steel walls begin to respond. The hull groans, the metal pops. At one point, there is a sharp crack loud enough to make both men flinch. For a moment, they do not know if the vessel has been breached. They check their instruments. Everything reads normal. One of the plexiglass windows in the outer structure had cracked, not the pressure hull itself, but the sound was indistinguishable from catastrophic failure. They kept descending. The Trieste was a bathyscaphe, which means it used gasoline for buoyancy, the way a blimp uses helium for lift. Gasoline is lighter than water and does not compress under pressure the way air does, making it an ideal float medium for deep dives. Iron shot pellets held in hoppers on the bottom of the vehicle provided ballast for the descent. To come back up, the crew would release the iron shot and let the gasoline carry them to the surface. Simple in theory, completely untested at those depths. At just over 32,000 feet, they hit the bottom. A puff of yellowish sediment bloomed up around the landing gear. The trench floor was real. They were sitting on it. And then Piccard looked through the porthole. There was something moving in the sediment. A flatfish, maybe a flounder-like species, perhaps a foot long, lying on the bottom and then slowly swimming away from the light of the submersible. At 32,000 feet, at pressure 1,000 times greater than the surface. A vertebrate animal, living and moving, and apparently untroubled by conditions that every previous scientific assumption said should be lethal. Walsh later said that seeing that fish was the single most surprising moment of the entire dive. They had expected the bottom to be barren. Instead, within minutes of arriving, they had observed complex animal life. Everything the scientific community had assumed about the deep ocean's habitability shifted in that instant. They spent 20 minutes on the bottom before releasing the iron ballast and beginning the long ascent back to the surface. The dive had proved something enormous, and it had also left most of the trench completely unexplored. The Trieste landed in one spot and saw one small patch of seafloor. The other 1,579 miles of the trench remained unseen, and what that unexplored territory eventually revealed made the fish look almost ordinary. Don Walsh saw it first. A pale shape, maybe a foot long, resting in the yellowish sediment at the deepest point ever visited by a human being. Then it moved. It pushed itself off the bottom with a slow, unhurried motion and drifted away from the light. For a few seconds, both men inside the Trieste watched a fish living its normal life at a depth that, according to every scientific textbook written before that moment, should have made life physically impossible. The scientific community did not fully accept this observation at first. The conditions inside the Trieste made it difficult to identify the fish with certainty. The single porthole was small, the light limited, and the sighting brief. Some researchers suggested it might have been a sea cucumber rather than a true vertebrate fish. The debate continued for years, but subsequent deep-sea missions eventually settled the question. Cameras dropped to extreme depths in the Mariana Trench have now captured fish on multiple occasions. The species most commonly documented is the snailfish, a translucent, eel-like animal that has become the confirmed deepest living vertebrate on Earth. Footage taken in 2017 showed them swimming actively at depths exceeding 26,000 feet, feeding on small crustaceans with apparent ease. A camera deployment in 2023 recorded a snailfish at approximately 27,000 feet, the deepest confirmed fish sighting in history at that time. The existence of fish at those depths completely changed the scientific framework for understanding the limits of vertebrate life. The vertebrate body plan, a spinal column, an internal skeleton, complex organs, a brain, was assumed to have inherent pressure limits. Bones crush, organ cavities collapse, neural tissue loses function. Every one of those vulnerabilities is real. The snailfish bypassed all of them through a combination of soft tissue, absent air pockets, and the TMAO chemistry discussed earlier. But the fish Walsh and Picard saw was almost certainly not a snailfish. The species they reported appeared to be a different type entirely, possibly a bottom-dwelling flatfish, adapted to live on sediment. Researchers today believe the animal likely belonged to a group called the Liparidae, but the exact identification remains uncertain. The trench was not yet a place where specimens could be collected easily, and an uncertain visual sighting from a tiny porthole in 1960 was the best evidence available. That gap between sighting and identification captures something important about the state of deep-sea science. Enormous creatures or organisms with enormous implications get spotted in passing and then take decades to study properly. The ocean is simply too large, too deep, and too difficult to access for science to move quickly. After Picard and Walsh surfaced, the scientific establishment began reorganizing around the new reality they had confirmed. The deep ocean was alive. The question shifted from whether anything lived there to how, and the answer to that question required understanding not just the biology, but the physics of what the descent itself does to the vehicle and everything inside it. Because the Trieste did not come back unchanged, the ocean had physically altered it during the dive, and that detail tells you something profound about the forces at play in the deepest place on Earth. The Trieste came back smaller, not dramatically, not in a way you could see with your eyes from a distance, but the pressure at the bottom of the Mariana Trench had been so immense that the steel hull of the bathyscaphe had physically compressed during the descent. The metal had deflected inward under the force of the water pressing against it from all sides. Engineers measured the deformation after the dive and confirmed it. The vessel had shrunken by a measurable amount. This is not how we normally think about steel. Steel is rigid. It holds its shape. You build bridges and skyscrapers from it because it does not flex. But at 16,000 pounds per square inch, even steel behaves differently. The atoms that make up the metal get pushed slightly closer together. The structure compresses. Then, when the pressure releases during ascent, the metal springs back. If the metal had been weakened by manufacturing flaws or fatigue cracks, the pressure could have caused a catastrophic failure. The Trieste's engineers had calculated the margin carefully, but those calculations were theoretical. The dive proved they were correct, but only barely. The styrofoam demonstration makes this concrete in a way the steel story does not. Researchers regularly attach styrofoam cups to equipment being lowered to extreme depths. Styrofoam is full of tiny air pockets. Air compresses dramatically under pressure. By the time the equipment reaches the bottom of the Mariana Trench, every one of those air pockets has been crushed out of the foam. The cup comes back up miniature, dense, and hard, squeezed down to roughly the size of a thimble. The plastic matrix that gave the cup its shape is still there. The space inside it is gone. The same principle applies to any air-filled structure sent to those depths. A sealed plastic bottle would implode on the way down, crumpling inward as the external pressure overwhelmed the air pressure inside. Research teams have demonstrated this with empty bottles sent on equipment lines, and the results come back looking like crumpled paper. Biological tissue responds differently depending on what it contains. Soft tissue, that is uniformly fluid-filled, like muscle, transmits pressure evenly in all directions and avoids being crushed. Cavities filled with gas, like lungs or swim bladders, collapse inward catastrophically. This is why deep-sea creatures at extreme depths have no air spaces in their bodies. Their anatomy has eliminated the one feature that pressure would destroy. The windows of the Trieste were another engineering challenge. The porthole that cracked during the descent was made from cone-shaped plexiglass. The design was intentional. A cone-shaped window, wider on the outside, gets forced more tightly into its seat as pressure increases. The pressure itself helps hold the window in place. The crack that occurred was in the outer structure, not in the main pressure hull, and it did not compromise the dive. But the sound it made convinced both men inside that they might be about to die. The engineering required to survive the trench is extreme by any standard, and the hardware built to explore it has improved enormously since 1960. But the trench itself has not changed. It remains as hostile and as crushing as it was when Piccard and Walsh first descended. Every vehicle, every sensor, every camera sent down there is fighting the same physics. And at the edges of the trench, the physics does something even more bizarre. It creates boiling water that never actually boils. Water boils at 212 degrees Fahrenheit. Every student learns this. Put water in a pot, heat it to 212, and it turns to steam. The liquid becomes gas. That is what boiling is, a phase change driven by heat. At the bottom of the Mariana Trench, hydrothermal vents blast water out of the sea floor at temperatures reaching 700 degrees Fahrenheit. That is more than 3 times the boiling point of water. At the surface, that water would vaporize in an instant. At the bottom of the trench, it stays liquid. It exits the vent as a super hot jet of liquid water, and it remains liquid the entire time. The reason is pressure. Boiling happens when water molecules gain enough energy to escape the liquid and become gas. Pressure from the surrounding atmosphere pushes back against that escape, which is why water boils at a lower temperature at high altitude, where the atmosphere is thinner. Less pressure means less resistance to the molecules leaving. More pressure means the molecules need more energy to escape. At the bottom of the Mariana Trench, the pressure pushing down on that water is 1,000 times what you experience at sea level. To boil under those conditions, water would need to reach temperatures far above 700 degrees. So the vent water, despite being incomprehensibly hot, has nowhere to go. The pressure holds it in liquid form. This creates a bizarre situation. Superheated liquid water jets out of the seafloor and mixes with near-freezing ocean water nearby. The temperature gradient in that mixing zone is some of the most extreme found anywhere on Earth, from nearly freezing to hundreds of degrees within a few feet. And in that zone, bacteria are living. Some bacteria that cluster around hydrothermal vents can tolerate temperatures of 250 degrees Fahrenheit or higher. These hyperthermophiles, organisms that love extreme heat, were essentially unknown before vent research began in the 1970s. Scientists assumed that 150 to 160 degrees was the absolute upper limit for life, based on what was known from surface biology. Vent research kept pushing that limit upward. The most heat-tolerant organism currently known to science was discovered in a deep-sea hydrothermal environment. It can survive and reproduce at temperatures approaching 300 degrees Fahrenheit. Its proteins stay functional at temperatures that would denature and destroy the proteins in your own cells in seconds. Hydrothermal vents also deposit minerals as the superhot water cools and reacts with the cold ocean. Over years and decades, those minerals build up into towering chimney structures called black smokers. The chimneys are named for the dark plumes of mineral particles they pour out, which look exactly like smoke from an industrial smokestack. Some black smoker chimneys reach heights of 60 feet or more, constructed entirely from mineral deposits that precipitated out of vent water. Those chimneys are ecosystems in their own right. Tube worms, clams, crabs, and thick bacterial mats coat their surfaces. The biodiversity around a single active black smoker rivals ecosystems found in temperate ocean environments with abundant sunlight. And the chemistry inside those vents is where scientists found something that completely changed the conversation about the origin of life. But that is only part of the story, because not all the vents in the trench push out hot water. Some of them push out something far stranger. The hydrothermal vents near the Mariana Trench do not all behave the same way. Most push out superheated mineral-rich water, but in a specific volcanic zone near the trench called the South Tonga Arc, and in other deep Pacific locations, researchers discovered something that required a second look at the footage to believe. The vents there push out liquid carbon dioxide. At the surface, carbon dioxide is a gas. You exhale it with every breath. It fizzes in your soda. It forms as wood burns. At surface pressure and room temperature, turning CO2 into a liquid requires chilling it to nearly -70 degrees Fahrenheit, or compressing it significantly. It does not occur naturally in liquid form in most environments on Earth. At depth, under extreme pressure, carbon dioxide can exist as a liquid at temperatures much closer to normal. The pressure forces the molecules together, keeping them in liquid form rather than allowing them to expand into gas. When a vent pushes carbon dioxide-rich fluid out of the seafloor under those conditions, the carbon dioxide emerges as liquid and settles onto the seafloor below the vent opening. Because liquid carbon dioxide is slightly less dense than seawater at those depths, it pools in low-lying areas of the seafloor rather than dispersing. Researchers filming these features described the scene as looking like a lake on the bottom of the ocean. The liquid carbon dioxide sat in a shimmering lens-shaped body with a distinct surface separating it from the water above. Rocks and sediment were visible through it, distorted by the difference in refractive properties between the two fluids. It looked, on camera, exactly like a pool of clear liquid sitting in a bowl on the seafloor. These carbon dioxide pools were studied for the organisms that inhabit their edges. The liquid carbon dioxide itself is hostile to life at high concentrations, but bacteria and other microorganisms were found living in the boundary zone where the carbon dioxide pool meets the surrounding seawater. They exploited the chemical gradient, using the energy difference between the two environments to drive their metabolism. The discovery added another entry to the list of conditions that life on Earth has learned to exploit. Every new extreme environment researchers investigate turns up organisms that have adapted to it specifically. The pattern is consistent enough that scientists have stopped being surprised when they find life somewhere previously assumed to be barren. Instead, they ask what the organism is using for energy and how its chemistry differs from surface life. The trench is full of these surprises, stacked on top of each other at every depth. And then there are the mud volcanoes, which push up material from deeper inside the Earth than any hydrothermal vent, material that carries traces of something scientists had not expected to find. Something that suggests life might not stop at the seafloor, something that implies the trench may be sitting on top of an entire hidden world that nobody has ever seen. Along the edges of the Mariana Trench, the seafloor has cracks. Through some of those cracks, material oozes upward in slow, thick flows. Scientists call these features mud volcanoes, but they behave nothing like the volcanoes you picture from a geography class. There is no fire, there is no lava, there is no dramatic eruption. Instead, cold, pressurized fluid works its way up through fractures in the crust, carrying sediment and dissolved chemicals with it, and emerges on the seafloor as a slow, steady seep of cold mud. The material that comes out is strange. It includes methane, hydrogen sulfide, brine, and fragments of organic compounds. Some of those organic compounds originated miles beneath the seafloor, carried up from depths far beyond where any drilling equipment has ever reached. Researchers analyzed the chemistry of the mud volcano output and found carbon signatures indicating the material had been sitting inside the Earth's crust for an extremely long time, subject to the heat and pressure deep inside the Earth before migrating upward through fractures. But the most unexpected part was what came with it: microbial life. Bacteria living inside the mud itself, in the fluids seeping through the cracks, using the chemical energy of the materials moving through the crust as their food source. These bacteria had not come down from the ocean above. Their chemistry and position indicated they had been living inside the rock, deep below the seafloor, for an unknown but potentially enormous period of time. This observation opened a question that scientists are still working to answer. How far down does life actually go? Scientists refer to this possibility as the deep biosphere, the idea that microbial life may extend miles beneath the surface of the Earth, living inside rock fractures, using chemical energy from geological processes, completely disconnected from the surface world and completely independent of sunlight. Research from multiple locations around the world has provided evidence supporting the existence of this deep biosphere. Bacteria have been recovered from rock samples drilled more than 2 miles below the seafloor in various ocean drilling programs. Near the Mariana Trench, the mud volcanoes push material up from approximately 6 miles beneath the seafloor. If microbial life exists in the rock at that depth, the total volume of living organisms inside the Earth's crust could be genuinely staggering. Some estimates suggest that the total mass of microbial life inside the Earth could rival the total mass of all life on the surface. Scientists are careful to call these estimates preliminary and heavily uncertain, but the idea is no longer treated as fringe speculation. The evidence is real. The implications extend past Earth. If life can colonize rock fractures deep inside a planet, surviving on chemical energy from geological processes, then the surface conditions of a planet become much less important for determining whether it can support life. A planet with a frozen surface and a warm, chemically active interior could be inhabited. Several rocky bodies in our solar system fit that description. And there is something even stranger happening at the edges of the trench's vent systems, something that reaches down into the chemistry of the rock itself and begins acting like a living thing. Something that, when scientists first described it, made the line between geology and biology almost impossible to find. In the deep ocean near certain hydrothermal systems, researchers found tiny structures built from minerals. They were nanometer scale, meaning smaller than 1/1,000,000th of a meter, under a powerful enough microscope. They looked almost biological, organized, layered with internal channels running through them. They were made entirely of rock, but they were doing something that should only be possible in living cells. They were converting energy. Living cells perform a process called ion transport. They move charged particles called ions across their membranes. This movement generates a difference in charge between the inside and outside of the cell, and the cell uses that charge difference to power its chemical reactions. It is one of the most fundamental processes in all of biology. Every living cell on Earth does it. Your own cells do it continuously to keep you alive. The mineral structures in the deep sea were creating the same kind of charge separation without any biological molecules at all. They were forming spontaneously from the chemistry of the vent fluid mixing with seawater, and their layered, channel-like architecture was allowing ions to pass through in a controlled way, generating osmotic energy, just like a cell membrane does. The researchers who published this finding were careful in their language. They described the structures as performing lifelike energy conversion, not as being alive. The distinction matters. A mineral structure that generates a charge gradient is not the same as a cell. It has no genetic material it does not reproduce. It cannot evolve. But the function it performs, the movement of ions to generate usable energy, is identical to what every living cell on Earth uses to power itself. This observation feeds directly into a major scientific debate about the origin of life on Earth. One prominent theory holds that life did not begin as a free-floating molecule in a warm puddle. Instead, it began at the interface between mineral surfaces and hydrothermal vent fluids in the deep ocean. The minerals provided the scaffolding. The chemical gradients provided the energy. Organic molecules accumulated in the channels of the mineral structures and began reacting. Over time, the chemical reactions became more complex. Eventually, the system enclosed itself in a membrane and began reproducing. Life emerged from the chemistry of rocks and water. If this theory is correct, then the mineral structures in the Mariana Trench area are not just curiosities. They are a snapshot of the process that produced the first living things on Earth, roughly 4 billion years ago. The conditions that exist at deep-sea hydrothermal vents today may closely resemble the conditions that sparked the origin of life itself. The evidence supporting this theory is not conclusive. The origin of life remains one of the deepest unsolved problems in science. But the discovery of these energy-converting mineral structures removed the biggest objection that critics raised against the hydrothermal vent origin of life theory. Critics had argued that vent chemistry lacked a mechanism for generating the energy gradients that life requires. The mineral ion channels answer that objection directly. The trench may be showing us where we came from. And if it is, then what that means for the possibility of life elsewhere in the universe is something that scientists are only beginning to work through. The hydrothermal vents near the Mariana Trench are producing something remarkable. And once you understand what they are doing, the origin of life on Earth stops seeming like a miracle and starts seeming almost inevitable. Here is the setup. Seawater seeps down through cracks in the seafloor. It gets heated by the rock below, picks up minerals, and rises back up through different cracks. When it hits the cold ocean water, the minerals precipitate out and build structures. Those structures have thin walls with tiny pores and channels running through them. The inside of the channel has one chemistry, the outside has another, and the gradient between them generates energy. That gradient is the key. All living cells use energy gradients to power themselves. A gradient is simply a difference, more of something on one side of a barrier, less on the other. Cells pump ions across their membranes to create these differences, then use the tendency of ions to flow back through to run their chemical machinery. It is the same principle as a dam. Water piles up on one side. When you let it flow through, you capture the energy of that flow. The mineral structures at deep-sea vents do this spontaneously. The vent fluid contains one set of chemicals. The ocean water contains another. The thin mineral walls separating them allow selective passage of certain ions. A gradient forms. Energy is available. Early Earth 4 billion years ago had no free oxygen in the atmosphere. The oceans were warm and chemically active. Hydrothermal systems were almost certainly operating along the ocean floor. The conditions for exactly this kind of mineral channel energy conversion existed in abundance. Organic molecules, the carbon-based building blocks of life, form naturally in hydrothermal environments. Amino acids, the components of proteins, have been synthesized in laboratory simulations of hydrothermal vent chemistry. Nucleotides, the building blocks of genetic material, have been produced in similar conditions. The raw materials for life were present. The energy gradients were present. The mineral scaffolding to concentrate and organize reactions was present. The theory proposes that early organic molecules began accumulating inside the mineral channels, where the energy gradient could power reactions between them. Over time, the reactions became self-sustaining. Molecules that catalyzed their own replication became more common. The system bootstrapped itself toward complexity. At some point, the whole arrangement enclosed itself in a lipid membrane, which is the fat-based boundary that surrounds every living cell today. That enclosure was the first true cell. Nobody watched this happen. The event is 4 billion years old and left no direct fossil record. But the chemistry is real, the conditions are consistent, and the mineral structures performing energy conversion in the Mariana Trench area today provide a working example of the kind of system the theory requires. If this origin of life pathway is correct, then life did not require lightning striking a puddle of chemicals or a lucky collision of molecules in the open ocean. It required a steady source of chemical energy, mineral scaffolding to organize reactions, and time. The deep sea provides all three in abundance. And if those conditions are sufficient, they are also common. Hydrothermal systems form wherever water meets hot rock in a planet's interior. They exist on ocean moons. They likely existed on Mars during its wet period. The recipe for life, if the vent theory is right, is available across the solar system. The trench may be the closest thing on Earth to a living laboratory for life's origins. And while scientists were working out that idea, a different kind of explorer was heading to the bottom for entirely different reasons. A filmmaker, alone, in a vehicle he helped design, and what he found there surprised even him. James Cameron made his name telling stories about the ocean. He directed a film about the Titanic that won 11 Academy Awards. He spent years studying the wreck personally, diving to it repeatedly in Russian submersibles. The ocean was not a backdrop for him, it was an obsession. So when he decided he wanted to reach the deepest point on Earth, he did not simply fund a mission, he helped engineer the vehicle himself. Cameron's submersible was called the Deep Sea Challenger. It was designed as a vertical torpedo, meant to drop straight down rather than glide. Traditional research submersibles are horizontal, shaped roughly like a bus. Cameron's team calculated that a vertical design would reduce drag during descent and cut travel time significantly. The vehicle was 12 feet wide and 24 feet tall. Cameron's personal compartment inside it was a sphere 43 inches in diameter. Imagine sitting inside a large exercise ball with your knees tucked and instruments surrounding you on every side. That was where he spent hours. On March 26, 2012, Cameron descended alone to the Challenger deep. The dive took roughly 2 hours and 36 minutes. He reached the bottom at a depth of approximately 35,000 feet. He became the first person to make a solo dive to the deepest point on Earth, and only the third person in history to reach it at all, after Piccard and Walsh more than 50 years earlier. He spent about 3 hours on the bottom before a hydraulic problem cut the mission short. He had planned for 6. The sample collection arm, designed to gather sediment and biological specimens, malfunctioned. The thrusters that were supposed to let him move laterally across the seafloor did not perform as expected. The sediment the vehicle disturbed during landing clouded his visibility for the first part of the bottom time. Despite the equipment problems, what he reported from the bottom was striking. The seafloor was flat, almost featureless, a vast pale expanse of soft sediment stretching in every direction to the limit of his lights. He described it later as feeling like the loneliest place on Earth. No fish were visible during his time on the bottom. No large organisms. Just the ooze, the cold, and the dark. He collected sediment samples before ascending. Those samples were later analyzed and found to contain microbial life, confirming that biology persists even in the compacted sediment at the very bottom of the trench. The microbes were slow-moving, low-energy organisms, adapted to cold and pressure, but they were alive and metabolically active. Cameron's dive drew global attention to the Mariana Trench in a way that purely scientific missions rarely achieve. Millions of people who had never considered the deep ocean watched footage from the bottom of the world. The scientific data he brought back was real and useful. The sediment samples contributed to ongoing research. But perhaps more importantly, his dive demonstrated that a solo human could survive the descent and return in a specially engineered vehicle, opening a technical pathway that other expeditions would follow. One of those expeditions came 7 years later. The person leading it set a new depth record, survived a descent deeper than any human before him, and within minutes of landing on the bottom, saw something that made the entire scientific case for the trench's isolation from human civilization collapse in an instant. Victor Vescovo is a retired United States Navy officer, a private equity investor, and an explorer. He had already climbed the highest peak on every continent and skied to both poles before he turned his attention to the oceans. His goal was to reach the deepest point in every ocean on Earth. He called it the Five Deeps Expedition. His submersible, called the Limiting Factor, was the most advanced deep-diving crewed vehicle ever built. Unlike the Trieste, which could only descend and ascend in a straight line, the Limiting Factor had full thruster control in all directions. It could hover, move laterally, spin in place, and hold position over a specific point on the seafloor. It carried 2 torpedo-shaped external modules packed with scientific equipment. Its crew sphere was made of titanium 90 mm thick. On April 28, 2019, Vescovo descended into the Challenger Deep and reached a depth of 35,849 feet. This broke the previous depth record and made him the deepest diving human being in history. He spent 4 hours on the bottom, longer than any previous crewed mission. During that time, his cameras captured the seafloor in high resolution. Scientists watching the footage later confirmed the presence of previously unrecorded features. New species of sea cucumbers and small crustaceans were observed. The sediment structure showed layering that gave geologists information about the history of sedimentation in the trench. And within minutes of landing, his cameras picked up something else entirely. A plastic bag. White. Intact. Sitting on the seafloor at the deepest point on Earth. Vescovo also observed what appeared to be candy wrappers and other manufactured debris resting in the sediment nearby. The items were not clustered. They were scattered, settled individually, exactly as they would look if they had drifted down slowly through 7 miles of water over an unknown period of time. Vescovo's reaction, documented in interviews afterward, combined frustration and resignation. He had traveled to the most remote location accessible to a human being and found evidence of the everyday human world sitting at the bottom of it. The bag had not been carried there by any intentional act. It had entered the ocean somewhere on the surface, drifted with currents, gradually sank as it became waterlogged, and descended through the entire water column to rest on the deepest floor on Earth. The discovery was not a complete surprise to marine biologists. Earlier research had already found synthetic fibers and microplastic particles at deep ocean depths, but the visual of an intact, recognizable grocery bag sitting on the Challenger deep seafloor communicated something that data tables could not. The reach of human pollution was total. There was no place left on Earth so remote that human waste had not arrived ahead of any human explorer. The bag on the floor of the Mariana Trench became one of the most referenced images in the conversation about ocean pollution. And it raised a question that the footage alone could not answer. How did so much human material end up at the bottom of the deepest place on Earth? And why can none of it ever leave? The Mariana Trench does not collect trash by accident. Its geography makes it almost inevitable. The trench sits in a basin surrounded by the western Pacific Ocean, one of the busiest and most plastic-polluted ocean regions on Earth. The Pacific Ocean contains an area called the Great Pacific Garbage Patch, a zone where rotating ocean currents, called gyres, concentrate floating debris. The garbage patch is estimated to cover an area roughly twice the size of Texas, and it contains an enormous mass of plastic fragments, ranging from large pieces down to microscopic particles. Ocean gyres move in slow, circular patterns driven by wind and the Earth's rotation. They act like drains, drawing floating material toward their centers. The material accumulates. Over time, the plastic debris in these gyres gets broken down by sunlight and wave action into progressively smaller pieces. Large plastic bags become medium fragments. Medium fragments become small chips. Small chips become particles invisible to the naked eye. As plastic breaks down, it becomes heavier relative to the water, and it begins to sink. Plastic also absorbs organic material from the surrounding water, which adds weight and encourages it to sink further. The particles drift down through the water column, passing through different depth zones over weeks, months, or years. The bottom of that sinking pathway for enormous amounts of material in the Western Pacific is the Mariana Trench. The trench is the lowest point in the region. Water flows downslope toward it along the seafloor. Particles carried by that flow accumulate at the bottom. The trench acts as a passive collector, a geographic endpoint for anything that sinks within its drainage area. Once material reaches the bottom, it stays. There are no currents at the bottom of the trench strong enough to lift heavy particles back up. The water at extreme depths moves extremely slowly, almost imperceptibly. Sediment settles and compacts. Plastic items sit on the surface of that sediment and remain. There is no mechanism that flushes the trench floor clean, no geological process operating on a human timescale that would remove debris once it has settled. Scientists studying the accumulation pattern of plastic in the deep ocean describe the trench as an ultimate sink. Material enters from the surface, sinks through the water column, and arrives at the seafloor with no pathway out. Every year that human plastic production continues, the inventory at the bottom of the trench grows. The rate of sinking varies. A small, dense plastic particle might reach the bottom of the Mariana Trench in a few weeks. A large, buoyant plastic sheet might take years to become heavy enough to sink at all, spending time floating at the surface and drifting thousands of miles before finally descending. This means the plastic arriving at the bottom of the trench today entered the ocean at many different times and from many different locations, some of them potentially thousands of miles away. The trench is a record of human waste going back decades, and the organisms living at the bottom have not been merely coexisting with that waste. They have been consuming it, and what scientists found inside their bodies changed the way researchers understood just how far into the food chain plastic pollution had already traveled. In 2019, a team of researchers published results from a study that stopped the marine biology community cold. They had collected amphipods from the Mariana Trench, the small shrimp-like crustaceans that live at extreme depths and form a major part of the deep-sea food web. The researchers examined the digestive systems of those amphipods under laboratory conditions. Every single one contained synthetic fibers or plastic fragments. Every single one. The amphipods were collected from depths exceeding 21,000 feet. They had never been near the surface. They had never come into contact with any human environment. Their entire lives existed in total darkness, under crushing pressure, 7 miles below the ocean surface. And their guts were full of plastic. The fibers found inside them included materials consistent with nylon, polyethylene, and polypropylene. These are materials used in fishing nets, food packaging, clothing fibers, and single-use plastic items. The researchers matched some of the fiber types to specific industrial applications. The composition of what they found was a cross-section of the global plastic economy in the digestive tract of an animal living at the deepest accessible point on Earth. The mechanism is straightforward. Amphipods are scavengers. They eat marine snow, the constant rain of organic material falling from the surface. Marine snow includes not just dead organisms and waste, but also the microplastic particles that have mixed into the upper ocean over decades. The plastic particles are similar in size to organic food particles. The amphipods eat them both indiscriminately. The plastic accumulates in their digestive tracts because biological systems have no mechanism for breaking down synthetic polymers. The implications extend upward through the food chain. Organisms that eat amphipods absorb the plastic their prey consumed. Organisms that eat those organisms accumulate even more. Each step up the food chain concentrates the plastic further, a process called biomagnification. By the time you reach the top of a food chain that began with microplastic-eating amphipods at the bottom of the Mariana Trench, the concentration of synthetic material in the tissues of the apex predator can be dramatically higher than what was present in the original source. Microplastics carry additional hazards beyond their physical presence. Plastic polymers absorb other chemical pollutants from the surrounding water. Pesticides, industrial chemicals, compounds that interfere with hormones. When an organism eats a plastic particle, it ingests the particle and all the chemicals the plastic absorbed during its time in the water. The plastic acts as a delivery mechanism for chemical pollution that would otherwise be dispersed too thinly in the water column to cause biological harm. The trench amphipods are at the bottom of the ocean, but they are part of a connected system. Their food comes from the surface. Their pollutants come from human industry. Their bodies tell the story of a food chain that stretches from plastic production facilities to the deepest living animals on Earth. And the sediment they live on tells a different kind of story. A story written in layers, over millions of years, in a material that looks like nothing much but contains a record of almost everything that ever lived in the ocean above it. Step onto the floor of the Mariana Trench, and your foot would sink into something soft, not sand, not rock, a thick yellowish-brown material that coats the entire seafloor of the deep trench. Scientists call it pelagic ooze, and it is one of the most information-dense substances on Earth. The ooze is made from the accumulated remains of hundreds of millions of years of ocean life. Every organism that lives in the surface ocean eventually dies. Its body sinks. Most of it gets consumed or decomposed long before it reaches the bottom, but a fraction survives the journey. The calcium-containing parts dissolve at depth because of the aggressive deep ocean chemistry. What remains is the organic material that resisted breakdown: silica structures from certain algae, organic molecules from cell membranes, fragments of carbon-based material from biological processes. Layer by layer over geological time, this material settles on the sea floor. The layer at the very top of the ooze represents recent decades. A few feet deeper is material from thousands of years ago. Deeper still, material from hundreds of thousands of years. At the deepest layers of the sediment in the Mariana Trench region, the record extends back tens of millions of years. Scientists extract this record by using long, hollow tubes pushed into the seafloor to collect sediment cores. A core pulled from the trench region is essentially a vertical timeline of the ocean's history. Researchers can identify the remains of specific species and determine when they appeared or went extinct. They can trace changes in ocean temperature through the chemical signatures preserved in the sediment. They can identify major geological events, like large volcanic eruptions, from layers of ash embedded in the ooze. The ooze also preserves a record of the Earth's magnetic field. Certain minerals in the sediment align with the magnetic field at the time they were deposited, and then lock in place as the sediment compacts. Because the Earth's magnetic poles have reversed multiple times in the past, flipping north and south, these magnetic signatures create a barcode-like pattern in the sediment layers. Scientists use this barcode to cross-reference sediment cores from different locations around the world and build a global timeline of ocean conditions. At the bottom of the trench, where the ooze has been accumulating for the longest time, the record is extraordinarily detailed. Organisms too small to see with the naked eye left chemical traces that survive for tens of millions of years. Events that happened before any human civilization existed are readable in the sediment, if you have the tools and training to interpret them. The ooze is also home to a living community. Bacteria thrive throughout the sediment column, using the organic material locked in the ooze as their food source. Their metabolic activity is extremely slow by surface standards. A bacterium in deep-sea sediment might divide once every few thousand years, but across the entire volume of sediment in the trench, the total metabolic activity adds up to something significant. Scientists studying this community believe it may represent a substantial fraction of the total microbial life in the ocean. The ooze that looks like nothing turns out to be a library, a habitat, and an archive of almost everything the ocean has ever produced. And lying on top of all of it, in the coldest and darkest place on Earth, are things that traveled there from the surface world entirely uninvited. things that will still be there long after every person alive today is gone. But the ooze is just the surface of the mystery. Beneath it, the answers only get stranger. You cannot see the bottom of the Mariana Trench. No light reaches it. Satellites that can photograph the surface of Mars to within a few feet of resolution are completely useless here. The water blocks everything. To map the trench, scientists had to invent a different approach entirely, and what those maps revealed when they were finally assembled surprised everyone who built them. The technique is called multi-beam sonar. A ship at the surface sends out pulses of sound in a fan-shaped array across the seafloor. Each pulse travels downward at roughly 1 mile per second through the water. When it hits the seafloor, it bounces back. The ship records how long each return takes, and from that timing, calculates the exact depth at thousands of individual points across the seafloor in a single pass. Move the ship forward and repeat. Stitch together thousands of passes, and you have a map. The resulting maps have a resolution that depends on water depth. In shallower water, sonar can resolve features as small as a few feet across. At Mariana Trench depths, the resolution drops to roughly 200 to 300 feet at best. That means any feature smaller than 200 feet across effectively disappears from the map. The trench is 7 miles deep, and the echo takes a long time to return, leaving room for error to accumulate. Despite those limitations, the maps produced unexpected details. The floor of the trench is not a simple flat basin. It has ridges, valleys, and localized depressions within the main depression. The Challenger Deep itself contains several distinct pools at slightly different depths, separated by low ridges of sediment. The deepest of those pools, where Vescovo set his depth record, is called the Western Pool. The walls of the trench drop steeply from the surrounding seafloor. In some places, the angle of descent exceeds 45 degrees. Landslides are common along the walls, triggered by earthquakes from the subduction zone below. Fresh landslide scars appear on the maps as bright, angular features against the smoother surrounding sediment. Scientists can estimate when a landslide occurred by looking at how much sediment has settled over the scar. The crescent shape of the trench shows clearly in the full map. It arcs gently for over 1,500 miles, following the curve of the plate boundary. The deepest sections are not distributed evenly along its length. The Challenger Deep at the southern end is by far the deepest feature, while other sections of the trench are considerably shallower. Researchers also used sonar to identify the mud volcanoes along the trench margins, the locations of known hydrothermal vents, and areas of unusual sediment texture that may indicate active seeps. The maps have guided subsequent dive missions to specific targets rather than requiring teams to search blindly across the enormous trench floor. After decades of sonar surveying, less than half of the Mariana Trench floor has been mapped at even the limited resolution that multi-beam sonar provides at those depths. The ocean is vast. Research ships are expensive to operate. Survey time is limited. The trench that has been visited by humans fewer times than the surface of the Moon has also been mapped less thoroughly than the surface of Venus. And what remains unmapped may contain features that would force another complete revision of what scientists think is possible down there. 12 human beings have walked on the surface of the Moon. All of them were part of the Apollo program. The last one stepped off the lunar surface in December 1972. No human has returned since. Fewer than that number have visited the bottom of the Mariana Trench. As of the most recent major expeditions, the total count of people who have descended to the Challenger Deep stands at a handful. Jacques Piccard and Don Walsh in 1960, James Cameron in 2012, Victor Vescovo in 2019, and again on subsequent dives. A small number of passengers who paid for seats on Vescovo's submersible during later commercial science dives. The total is around 20 people, and the count grows slowly because the technical and financial barriers remain enormous. The Moon is 238,000 miles away. The Challenger Deep is 7 miles down. By any measure of physical distance, the ocean floor should be easier to reach. But distance is not the challenge. The challenge is the environment. The moon has no atmosphere and extreme temperature swings, but it has no pressure. A spacesuit protects an astronaut from vacuum and temperature. The suit is rigid and bulky, but the engineering is manageable. The moon does not actively try to crush the vessel carrying you there. The Mariana Trench pushes back. Every inch of descent adds more force to the structure carrying you. By the bottom, the vehicle is holding back the equivalent weight of 50 jumbo jets, pressing against every square inch of its hull. A single structural flaw, a microscopic crack in the titanium sphere, a failed seal around a viewport, means immediate and catastrophic flooding. There is no pressure suit that can protect a human body from that. The only protection is the hull itself, building a hull strong enough to handle that load, light enough to sink at a reasonable rate, and precise enough that no flaw exists in its construction, is genuinely difficult engineering. The limiting factor, Vescovo's submersible, cost tens of millions of dollars to design and build. It required years of engineering work from specialists in deep-sea pressure vessel design. Before any crewed dive, it conducted multiple unmanned test dives to verify structural integrity at depth. The result is that crewed visits to the bottom of the Mariana Trench happen at a rate of perhaps a few per decade. Robotic visits happen more frequently. Unmanned landers, cameras, and sampling equipment can be built to withstand pressure without the safety margins required for human life, which makes them cheaper and more practical. The Japanese robotic submersible Kaiko reached the bottom in 1995 and collected samples that confirmed microbial life. Other unmanned systems have followed. But robots observe what they are programmed to look for. A human scientist on the seafloor notices things, reacts to unexpected detail, decides in real time to move towards something unusual. The combination of rarity and human observation capacity means that every crewed dive to the Challenger Deep has returned with discoveries that unmanned missions missed or could not fully capture. The trench floor remains one of the least visited places on Earth, and each visit reveals that what we thought we knew about it was incomplete. The next discovery always comes from the thing nobody expected to see, which raises an uncomfortable question. If something large and biologically significant were living in the unexplored sections of the trench, would we even know yet? The Mariana Trench is seismically one of the most dangerous regions on Earth. This is not incidental to its existence. The process that created the trench, the subduction of the Pacific Plate beneath the Philippine Sea Plate, is the same process that generates earthquakes. The trench and its seismic activity are inseparable. Subduction zones produce earthquakes in two main ways. The first involves the interface between the two plates. As the Pacific Plate grinds beneath the Philippine Sea Plate, it does not slide smoothly. The two plates lock together at points of contact. Stress builds up over years or decades as the plates continue pushing against each other while the locked sections resist movement. Eventually, the stress exceeds the friction holding the plates together, and the locked section suddenly releases. That sudden movement sends seismic waves radiating outward through the Earth's crust in all directions. These interface earthquakes can reach enormous magnitudes. The subduction zone off the coast of Japan, which operates by the same mechanism as the Mariana Zone, produced the magnitude 9 earthquake in 2011 that generated the devastating tsunami that struck northeastern Japan. That earthquake released energy equivalent to roughly 9,000 atomic bombs detonating simultaneously. The death toll exceeded 15,000 people. The second earthquake mechanism in subduction zones involves the descending plate itself. As the Pacific Plate bends downward and sinks into the mantle, it experiences enormous internal stress from the bending. The rock is not flexible enough to bend without cracking. Earthquakes generated inside the descending plate, called intraplate or slab earthquakes, can occur at depths of hundreds of miles below the seafloor. These deep earthquakes feel different at the surface than shallow ones, and do not generate tsunamis as effectively, but they can still cause significant damage at the surface. Tsunamis generated near the Mariana subduction zone travel outward in all directions across the Pacific. The speed of a tsunami in the open ocean depends on water depth. In deep water, these waves travel at roughly 500 miles per hour, similar to the cruising speed of a commercial jet. They are not dramatically visible in the open ocean because their height is small relative to their wavelength. A tsunami wave in deep water might only be a foot or two tall, but could be hundreds of miles from crest to crest. It is only when it reaches shallow coastal water that it slows down, compresses, and rises to devastating heights. The Hawaiian Islands sit in the path of tsunamis that originate in the Western Pacific. Japan, the Philippines, Papua New Guinea, and numerous Pacific Island nations face regular tsunami risk from subduction zone activity in the Mariana region. Warning systems in the Pacific have improved significantly since the 1960 Hilo tsunami and the 2004 Indian Ocean disaster. Sensors on the seafloor detect the pressure signature of tsunamis and transmit warnings, but warning time for islands close to the source can be measured in minutes rather than hours. The trench generates hazards that reach far beyond its walls. It is a geological force with global consequences, and its activity is ongoing. Every year, dozens of earthquakes originate in the subduction zone beneath it. Most are too small to feel at the surface. Some are large enough to close beaches and trigger evacuations across the Pacific. The trench has never stopped moving, and it never will on any timescale that matters to a human life. The seafloor at the bottom of the Mariana Trench is approximately 180 million years old. That number places it among the oldest ocean crust on Earth. Most ocean floor gets recycled before it reaches that age. The process of subduction pulls seafloor into the mantle, where it melts and eventually gets recycled back to the surface through volcanic activity. New ocean floor forms continuously at mid-ocean ridges, where two plates pull apart and magma wells up to fill the gap. The global ocean floor turns over on a timescale of roughly 200 million years. The crust at the bottom of the Mariana Trench survived this recycling because it sits at the leading edge of the Pacific Plate, always advancing, always being pushed into the subduction zone, but never quite having arrived yet. It is the oldest oceanic crust that will ever be accessible to scientific study, because once it crosses into the subduction zone, it disappears into the mantle forever. That old rock contains chemical information that younger seafloor does not. The minerals in the crust locked in chemical signatures at the time they solidified, recording the composition of the ancient ocean and the ancient atmosphere. Some of those signatures tell scientists about the oxygen content of the early ocean. Others record the temperature of the water when the rock formed. Still others preserve information about the magnetic field, which has flipped dozens of times in the past and left a barcode of magnetic reversals in the rock. Reading this record requires collecting rock samples from the trench walls, which is a technical challenge. The walls are steep and unstable, prone to landslides triggered by the constant seismic activity below. A robotic arm reaching for a rock sample from a submersible risks triggering a slide that could bury the vehicle. Researchers approach these collections carefully, planning the sampling strategy based on sonar maps of the wall before any vehicle goes near it. The sediment above the old crust tells a different story. The layers of ooze accumulating over millions of years recorded biological events in the ocean above. Mass extinction events show up in the sediment as sharp boundaries where the fossil record of certain species simply ends. The extinction of the dinosaurs 66 million years ago left a chemical signature in deep ocean sediment worldwide, including in cores from the Mariana region. A thin layer enriched in the element iridium, which arrived from the asteroid impact that killed the dinosaurs, appears at the same depth in sediment cores from opposite sides of the planet. The trench floor is simultaneously the oldest and most historically complete archive in the ocean. The rock below preserves the chemistry of an ancient world. The sediment above preserves the biological history of the ocean that covered it. Together, they form a record stretching from the present day back through nearly 200 million years of Earth history. Scientists have barely begun to read it. The technical difficulty of collecting samples from the trench walls and the sheer volume of sediment covering the floor mean that most of this archive remains untouched. What has been read so far has already rewritten chapters of Earth history. What remains unread may hold answers to questions that scientists have not yet thought to ask. Along the margins of the Mariana Trench, in the transition zone between the steep trench walls and the surrounding seafloor, a different kind of geological feature operates quietly in the dark: cold seeps. They look nothing like the dramatic black smoker chimneys of hydrothermal vent systems. They produce no plumes, they generate no dramatic temperature contrasts. They seep methane and hydrogen sulfide, along with brine and other chemical-rich fluids, ooze upward slowly through fractures in the seafloor. The source of these fluids is deep in the sediment or in the rock beneath, where organic material buried under pressure over millions of years has broken down into simpler chemical compounds. Those compounds migrate upward through the porous sediment and fractured rock until they reach the seafloor and escape into the water column. The methane in particular is important. Methane is a powerful energy source for certain bacteria. Where methane seeps from the seafloor, communities of methane-oxidizing bacteria establish themselves in dense mats on the sediment surface. These bacteria, called methanotrophs, consume the methane and convert it into organic carbon that other organisms can use. They are the base of the cold seep food chain. Around these bacterial mats, a full ecosystem develops. Tube worms cluster in the chemically rich sediment. Clams with specialized gill structures that host methane-oxidizing bacteria inside their own tissue live in large aggregations. Small crabs pick through the mats. Predatory fish visit from the surrounding water column. The productivity of a cold seep ecosystem rivals that of a hydrothermal vent, despite operating at near freezing temperatures with no heat source. Cold seeps also produce a distinctive geological feature called orthogenic carbonates. As bacteria consume methane, the chemical reactions they drive cause calcium carbonate to precipitate out of the surrounding water and solidify in the sediment. Over years and decades, this builds up into hard carbonate rocks that protrude from the sea floor. These rocks can persist for thousands of years after a seep goes inactive, preserving a chemical record of past seep activity that scientists can date and analyze. The methane at deep cold seeps near the Mariana Trench arrives from depths that challenge geologists. Some of the carbon isotope signatures in the seeping methane indicate it originated from organic material buried 6 miles or more below the seafloor, far deeper than any cold seep would be expected to draw from. This implies the existence of deep fluid migration pathways connecting the surface of the seafloor to the deep interior of the crust, channels through which material moves upward over long periods. These same pathways may be the conduits for the deep biosphere organisms discussed earlier, microbes living miles below the seafloor that could migrate upward through cracks and seeps. The cold seeps near the trench may not be just local ecosystems. They may be windows into a much larger biological system operating inside the Earth itself. And if life can travel through rock fractures from miles beneath the crust to the ocean floor, the concept of where life lives on Earth needs to be fundamentally expanded. The trench is not a destination. It may be a crossroads between the surface world and something much deeper that we have barely begun to detect. Scientists studying the Mariana Trench have spent decades uncovering the ways life survives without sunlight, without bones, without oxygen at the surface, and under pressure that crushes steel. Each discovery has expanded the known range of conditions that life can tolerate, and each expansion has changed the calculation for where life might exist beyond Earth. Europa is one of Jupiter's moons. It is slightly smaller than Earth's moon and covered entirely in a layer of ice. Beneath that ice, confirmed by gravitational measurements and observations of the moon's magnetic field, is a global ocean of liquid water. The ocean is estimated to be between 40 and 100 miles deep, kept liquid by tidal heating from Jupiter's enormous gravity flexing the Moon's interior. The seafloor of Europa's ocean is in contact with a rocky mantle, and the conditions there may include hydrothermal activity similar to what operates along the Mariana Trench. Europa's ocean has never seen sunlight. Photosynthesis is impossible there. If life exists in that ocean, it runs on chemical energy from geological processes, exactly the way life runs in the deepest zones of the Mariana Trench. The organisms that evolved in the dark ecosystems of Earth's deep ocean are the closest known biological analogues to what life on Europa might look like. Enceladus, a moon of Saturn, is smaller than Europa, but perhaps even more compelling. Observations by the Cassini spacecraft confirmed that Enceladus vents water vapor into space from cracks at its south pole. That vapor contains organic molecules and silica particles, consistent with hydrothermal activity at the interface between the Moon's ocean and its rocky core. The chemistry in those vents is strikingly similar to what is observed in deep-sea hydrothermal systems on Earth. If the hydrothermal origin of life theory is correct, and if Enceladus has been maintaining the right chemical conditions in its subsurface ocean long enough, then life may have originated there by the same process that scientists believe sparked life in Earth's deep ocean billions of years ago. Titan, another Saturnian moon, has a completely different kind of ocean, lakes of liquid methane on its surface rather than liquid water. The chemistry there is wildly different from anything on Earth. But the discovery of life using methane as an energy source in cold seep environments near the Mariana Trench has opened the theoretical possibility that chemistry built around methane rather than water could support biology in ways scientists had not seriously considered before the deep sea revealed methane-based ecosystems. The Mariana Trench is rewriting the requirements for life. Sunlight is optional. Oxygen at the surface is optional. What remains required, based on everything known so far, is liquid water, a chemical energy source, and time. The trench demonstrates that those three conditions alone are sufficient to support extraordinary biological complexity. The universe contains enormous numbers of bodies with subsurface liquid water. The trench suggests that biological potential may be far more widespread than the surface conditions of any planet would indicate. What we assumed was rare turns out to be almost inevitable given the right basic ingredients. And in the trench itself, something is still being discovered that challenges one of the most basic assumptions biologists have ever made about what it means to be alive. Biology textbooks define life by a set of shared properties. Living things use energy, they grow, they respond to their environment, they reproduce, they maintain their internal chemistry separate from the outside world. Every organism ever officially classified as alive shares all of these properties. The definition seems solid. It was built over centuries of observation across millions of species. The Mariana Trench is making that definition wobble. The abiotic mineral structures performing ion-based energy conversion represent one challenge. They are not alive, but they perform a function that biology requires for life. The deep biosphere microbes push another boundary. Organisms so slow-moving and so deeply buried that their reproduction rate is measured in thousands of years. Are they alive in any meaningful sense, or do they sit at the very edge of the definition, barely qualifying? The viruses that live in the deep ocean sediment of the trench raise their own questions. Viruses are typically excluded from the definition of life because they cannot reproduce without hijacking the cellular machinery of a true living cell. They are obligate parasites of biology. But viruses in the deep ocean sediment exist in numbers that dwarf the bacteria living alongside them. Some researchers estimate that deep-sea sediment contains more virus particles than any other environment on Earth. These viruses infect and kill bacteria, regulating the microbial community and influencing the rate at which organic material in the sediment gets processed. They are not alive by the standard definition, yet they control the ecology of the deepest living system on Earth. The single-celled organisms at the bottom of the trench occupy a different uncomfortable space. The xenophyrophores, those softball-sized single cells, blur the line between unicellular and multicellular life. They contain thousands of nuclei sharing a single cellular boundary. Is this one organism or a colony? More recently, researchers studying deep-sea vent environments found evidence of viruses infecting archaea, the ancient single-celled organisms that dominate the deepest biological communities. These viral infections sometimes result in the transfer of genetic material between host cells, a process that accelerates evolution by moving useful genes between organisms that are not directly related. This horizontal gene transfer is a major mechanism driving the rapid adaptation of deep-sea microbes to their extreme environments. The viruses driving this process are not alive, but they are shaping the evolution of organisms that are. The deeper scientists go into the Mariana Trench's biology, the more the clean categories that biology relies on start to fray. Life and non-life are not always clearly separated. The energy-converting minerals look like early life. The viruses regulate living communities. The slow metabolizing bacteria test the lower limit of what we call living. The giant single cells challenge our picture of what an organism is. This does not mean the definition of life is wrong. It means the definition was built from studying the easily observable, moderately accessible life at the surface of Earth. The trench is showing us the margins, the edges where life starts and stops, where chemistry becomes biology, where the rules get complicated. Understanding those margins turns out to matter enormously, because everything we think we know about where to look for life in the universe was built on the same limited surface-based definition that the trench is now forcing scientists to revise. Evolution normally works slowly. A species adapts to its environment over thousands to millions of generations. Small changes accumulate, the individuals best suited to their conditions survive and reproduce. Their traits spread. Eventually, the population looks different from what it started as. The process is measured in geological time. In the Mariana Trench, evolution operates under pressure, quite literally, and the effects appear in the biology of deep-sea organisms in ways that tell scientists how fast extreme environments can drive adaptation. The clearest evidence comes from comparing the chemistry of closely related species living at different depths. A snailfish living at 5,000 feet and a snailfish living at 25,000 feet belong to the same broad family. Their body plans are similar, their basic biology is similar, but at the molecular level, they differ significantly. The deep-living snailfish has higher concentrations of TMAO, different enzyme structures, softer skeletal tissue, and cell membranes with higher concentrations of unsaturated fatty acids. These are not minor variations. They are fundamental changes to the chemistry that runs the body. For those changes to have occurred, natural selection had to work on many generations of organisms living at progressively deeper depths. Any individual whose TMAO concentration was slightly higher had a survival advantage. Any individual whose membrane fats were slightly more unsaturated was more likely to survive the cold. Over many generations, these advantages accumulated. The population shifted. The deep-living lineage diverged from its shallower relatives. The horizontal gene transfer driven by viruses in deep-sea sediment may accelerate this process. When a virus infects a deep-sea microbe and carries genes from one cell to another, it can transplant an adaptation that took thousands of generations to evolve in one lineage, directly into another lineage. The receiving organism may gain a pressure resistance adaptation in a single generation that its evolutionary line never would have developed independently. This mechanism dramatically speeds up the spread of useful adaptations through a population. The physical isolation of the trench also contributes to rapid divergence. The walls of the trench act as boundaries. Organisms adapted to the floor of the Challenger Deep cannot easily migrate to other ocean environments, and organisms from those environments do not easily enter the trench. The population at the bottom is genetically isolated, which means any mutations that arise there either spread through that isolated population or disappear. Isolated populations evolve more rapidly than connected ones because the genetic pool is small and selection pressure is intense. Scientists studying deep-sea amphipod populations from different trenches around the world found that populations from the Mariana Trench are genetically distinct from populations in other Pacific trenches. Despite the amphipods being similar species, the isolation of each trench has driven the populations toward different genetic solutions to the same extreme conditions. Same problem, different solutions, each shaped by whichever mutations happen to arise in that particular isolated population. The trench is not just an environment where life survives, it is an environment where life changes fast, pushed by pressures that have no equivalent anywhere on the surface. And those changes accumulate over millions of years, producing organisms that are genuinely unlike anything in the accessible ocean above. Some of those organisms may hold biological molecules or processes with applications that scientists have not yet imagined. Others simply exist as proof that life finds more paths than any single theory of evolution can predict. And the trench holds one final secret that brings all of this back to a single, startling conclusion about how much we still do not know. The deep biosphere is the name scientists give to a world within a world, a biological system operating inside the crust of the Earth, below the seafloor, beyond the reach of any drill currently deployed. Evidence for its existence has been accumulating for decades, and the picture that evidence is building is extraordinary. Drilling programs that extracted rock cores from several miles beneath the ocean floor have recovered living bacteria from the rock itself. These bacteria are not contamination from the drilling process. Their chemistry is distinctive. Their metabolic signatures indicate they have been living in the rock for an enormous period of time, and they cannot survive in the surface conditions that drilling equipment is assembled in. They are indigenous to the deep rock. The methane rising through cold seeps near the Mariana Trench carries carbon isotope signatures that indicate a biological origin from sources approximately 6 miles below the seafloor. Something was alive down there, consuming organic material and producing methane. And the methane is now migrating upward through cracks in the crust to emerge on the seafloor. The biology is inaccessible, but its chemical products are not. Estimates of the total volume of the deep biosphere vary widely because direct sampling is so difficult. But some researchers calculate that the total mass of microbial life inside the Earth's crust could be comparable to the total mass of all life on the surface of the planet. If those estimates are even partially correct, the majority of life on Earth exists somewhere humans have never seen, cannot easily reach, and have barely begun to study. The organisms in the deep biosphere live at rates that are almost incomprehensible by surface standards. A bacterium in surface soil divides every few hours. A bacterium in deep-sea sediment might divide once every 1,000 years. A bacterium deep in the rock might divide even less frequently, essentially existing in a state of chemical neostasis, repairing its genetic material just fast enough to prevent fatal mutations, consuming just enough chemical energy to maintain itself. This is life at its absolute minimum functional rate. These organisms have been isolated from the surface world long enough to develop genetics that are deeply divergent from surface life. Some of the archaea recovered from deep ocean drilling are classified in lineages that split from the main tree of life at such an early point that they provide information about the very first organisms to evolve on Earth. Studying them is, in a sense, studying the ancestry of all life on the planet. The Mariana Trench is a window into this hidden world. Its mud volcanoes bring material from depth to the surface. Its cold seeps vent the chemical products of deep biological activity. Its sediment columns preserve genetic material from organisms that existed millions of years ago. The trench is not the bottom of the ocean. It is the top of something much deeper and much larger than anyone had imagined when the HMS Challenger first dropped a weighted rope over the side and recorded a depth that nobody believed. There is a world beneath the world. It is alive. It has been alive for billions of years. It has survived every mass extinction that killed off the surface life above it, because it is deep enough that nothing from the surface can reach it. Not asteroids, not ice ages, not anything, except, now for the first time in the history of life on Earth, one thing from the surface has reached it: plastic. Everything covered so far represents what scientists have confirmed, detected, theorized with evidence, or directly observed, and it is extraordinary. A 7-mile deep canyon, creatures running on chemical energy, a single cell the size of a softball, bones replaced by sand, rock formations acting like living cells, a grocery bag at the deepest point on Earth, an entire biosphere living inside the planet itself. All of that is the known portion of the Mariana Trench. The unknown portion is larger. Less than 1% of the deep ocean floor has been directly observed by cameras or human eyes. The Mariana Trench stretches over 1,500 miles and has been visited in detail at only a handful of specific points, mostly clustered around the Challenger Deep at its southern end. The vast majority of the trench floor has never been filmed. Many sections have never been mapped at high resolution. Large areas have only been characterized by the water chemistry above them, with no direct observation of what lives there. Every single crewed or robotic dive to a new location in the trench has returned with organisms or features that were not expected. The snailfish population was more abundant and more diverse than predicted. The sediment microbial communities were more metabolically active than models suggested. The hydrothermal systems produced chemistry that did not match any prior template. The abiotic mineral structures performing energy conversion appeared without precedent in any earlier research. The deep biosphere signals grew stronger with every new drill core. The pattern is consistent. Every time a camera reaches a new section of the trench, something surprising is there. The expectation has shifted. Scientists no longer predict emptiness and treat life as the surprise. They predict life and treat its specific form as the variable to be discovered. That shift in assumption reflects how thoroughly the trench has overturned the original framework. The technology for exploring the trench is improving rapidly. Autonomous underwater vehicles, robotic systems that can navigate independently without a tether to a surface ship, are becoming capable of operating at full ocean depth. They can cover far more seafloor than a crewed submersible in the same amount of time. Future generations of these vehicles may be able to map and observe sections of the trench that are currently completely inaccessible. Swarms of small autonomous vehicles communicating with each other could theoretically survey the entire trench floor within a few years, if deployed. Even so, observation is not the same as understanding. Seeing an organism on camera is different from collecting it, sequencing its genome, analyzing its chemistry, and integrating that information into the broader scientific framework. The trench is so far from the surface and so expensive to access that the gap between what can be observed and what can be fully studied is enormous. Sample collection at full trench depth remains a major challenge. Specimens brought back alive are rare. Most biological material arrives at the surface already dead, partially degraded by the pressure change during ascent. The Mariana Trench is the most extreme environment on Earth that biology has colonized. It is the place where the rules of surface life break down completely and where a completely different set of rules takes over. Understanding those rules in full will take decades more of research, better technology, and continued willingness to be surprised. Every assumption that has gone into the trench has come back changed. That is the most reliable thing scientists know about it. 7 miles down, where no light has ever reached, where the weight of the ocean would kill you before you registered what was happening, something remarkable is occurring. Life is running the show. Life that does not need the sun. Life that turned rock into its energy source. Life that replaced its bones with sand and packed its cells with antifreeze. Life that has been living inside the planet itself for billions of years, surviving every catastrophe the surface has ever faced. The Mariana Trench started as a geological feature, a scar created by the collision of two tectonic plates and became the most consequential piece of ocean real estate on the planet for understanding what life actually is, what it requires, and how far it can go. Every assumption science brought to the trench was overturned. Life needs sunlight. Wrong. Organisms need bones to be complex. Wrong. The deep ocean is a barren desert. Wrong. The origin of life required a warm, sunny pond. Possibly wrong. The farthest point from human civilization is pristine and untouched. Definitively wrong. What the trench says instead is this: life is opportunistic, chemically creative, and nearly impossible to stop once it starts. Given a source of energy, no matter how unusual, given liquid water, and given time, life builds something. It builds single cells, the size of softballs. It builds colonies of tube worms around jets of 700-degree water. It builds ghost fish swimming freely at the deepest depth any vertebrate has ever survived. It builds bacterial communities inside rocks that have not seen the surface world in millions of years. And it does all of this in total darkness, under crushing pressure, at near-freezing temperatures, in a place that most people on Earth will never think about, and that fewer humans have visited than have stood on the moon. The trench also says something uncomfortable. Human civilization has left its mark on the most remote environment on Earth before we finished understanding what was there. The plastic bag on the Challenger deep seafloor arrived before most of the scientific story we have been telling was even discovered. Whatever biology lives in the sections of the trench not yet explored, whatever chemistry operates in the deep biosphere beneath it, whatever record is preserved in the 180-million-year-old seafloor rock, we have already contaminated the environment containing those answers. The trench will continue to exist long after every structure humans have ever built has been recycled by geological time. The subduction zone will keep pulling the Pacific plate downward. The hydrothermal vents will keep pushing hot water and minerals out of the seafloor. The bacteria in the rock will keep their slow, near-frozen metabolism running. The ooze will keep accumulating, layer by layer, preserving whatever is happening at the surface of the ocean in a record that will be readable millions of years from now. We visited. We found that the deepest, most hostile, most extreme place on Earth is alive, complex, and connected to the rest of the planet in ways we are still tracing. We found our own trash there before we found most of its species. And we left with more questions than we arrived with, which is exactly what happens every single time science reaches a new frontier. The trench is not the end of the ocean. It is the beginning of what we do not know yet. And everything we have found there suggests that what we do not know yet is bigger than everything we do.